Namaskaram. Welcome to KNA, Kerala NEET Academy. Okay. Today I am going to discuss some questions from the different chapters in the biology. Okay. So the chapters like cell cycle, biomolecules, and followed by molecular biology. Okay. These are chapters. There are some important areas. Okay, the areas which you may neglect, okay, those areas I am going to just recollect. The persons who are going to watch, definitely more or less, which is useful to you. Okay, this is the last duration towards exam, na? So whatever we are doing, it should be useful to the exam. Okay, so I am going to start with some questions here. So here one time you can check some questions. I am going to start with the chapter cell. Okay. The first question you are seeing on the screen. The following are the functions of mesosomes. Okay. This one, about the mesosome, that concept, the functions are important for the NEET exam. Okay, na? So before going to tell, what are the mesosomes? Mesosomes are some characteristic features present in the bacteria, or mesosomes are the characteristic features present in the prokaryotes. Indirectly meaning is what? Mesosomes such as structures not present in the eukaryotes. Okay, one. So these mesosomes, for example, when you are speaking about bacteria, we need to speak about the cell wall. Okay, within the cell wall, there is a cell membrane. Correct, na? These are cell membrane like a foldings, just like what it called as a sphericals, or in the form of tubules, or in the form of lamellae. Just first observe the mesosomes. First point is what? Mesosomes are the characteristic features of the prokaryotes or bacteria. One. Second point is that mesosomes structurally, these are the foldings of the plasma membrane. These are foldings of the plasma membrane in the form of vesicles, or in the form of tubules, or in the form of lamellae. Okay. This containing, these ones are the mesosome. Comes to their functions. Okay. Functions are important for the exam point of view. Okay. This one is a mesosomes function like they are involved in the help in the cell wall formation. Exam important mesosome function, cell wall formation. Second one, help in cell wall formation, help in the DNA replication. Helps in the replication. After DNA replication, one DNA molecule is there in bacteria, it becomes a 2 DNA molecule. After that, distribution to the cells. Distribution to the cells. Okay, that one important. The next one, very important thing is that respiration. Respiration. This point understand, okay? Respiration. Glycolysis, Krebs cycle, electron transport system comes under respiration, followed by the secretion. Now I am going to confirm the first point, that mesosome function. Mesosomes, what you should know. Okay? The function wise, mesosomes are help in the cell wall formation, helps in the DNA replication and distribution to the daughter cells, helps in the respiration and help in the secretion. These are the functions of mesosome. It is the same question, already I mentioned with reference of this one time you can check. Respiration. For example, what it called as if a same question if I frame in another way, which of the following is equals to mitochondria in the prokaryotes? You understand me? Which of the following is equals to, functionally which is equals to mitochondria in prokaryotes? Okay. What is the function of First, mitochondria present only in the eukaryotes. First, that is the point. What is the function of the mitochondria? Mitochondria is a site of aerobic respiration. Krebs cycle, electron transport system together called as a respiration. Thereby mitochondria generates the energy. So that's a normal. Which one is equals to mitochondria in prokaryotes, in bacteria? That's the answer is a mesosome, because mesosome function is a site of respiration. So here E 1 point. Okay, now this is altogether considered as a 1 point about mesosome. Same here, what is the definition of the mesosomes? I said mesosomes are the foldings of plasma membrane towards the cytoplasm or into the cell, towards the cytoplasm. These mesosomes are observed in the most of the bacteria. I am telling, why I will tell you, most of the bacteria you can observe the mesosome. Now comes to the, there is another one is called as a chromatophores. Some students having a small confusion between the mesosomes and the chromatophores. Chromatophores also same, this is a cell wall, this one is a cell membrane. These are also foldings of the cell membrane only. But these foldings are, this is mesosomes are the foldings of plasma membrane, chromatophores also foldings of the plasma membrane. This is present towards the cytoplasm, these are also towards the cytoplasm. The difference is mesosomes foldings containing a respiratory enzymes, already I mentioned. These chromatophores, these containing a pigments. Mesosomes are present in the most of the bacteria, but chromatophores are present only autotrophic bacteria. What is example of autotrophic bacteria? You can call it as a cyanobacteria, or you can call it as a blue-green algae. Better you can call it as a cyanobacteria. Okay, so difference you understand. So mesosomes are the foldings of plasma membrane, chromatophores are also foldings of plasma membrane towards the cytoplasm, but present in the most of the bacteria containing respiratory enzymes, containing a pigments present only in cyanobacteria only. Then what is the function of these chromatophores? These chromatophores are involved in the, directly I can tell, photosynthesis. Most correct is light reactions of photosynthesis. That is why here first one is first question you should make the difference between the mesosomes and chromatophore. There is a possibility of question based on the mesosomes and chromatophores. Now first question, I hope you are get cleared. What are the functions of mesosomes except cell wall formation, DNA replication, photosynthesis and respiration? I am going for option 3. There is no confusion about the answer. There is always confusion about the concept. Second one is that among the following structures in bacteria is or not locomotory. Directly we can tell. Normally we can tell. While you are reading the bacteria, locomotory, most common locomotor. For example, when you are calling as a locomotory structure, cilia will come, flagella will come. Yes or no? But even though both options are there, cilia and flagella, both options are there. Which one is the most common locomotory structure in bacteria? If somebody asks, go for the flagella. Understand the frequency. Okay? Both cilia and flagella options are there, locomotory structures. But you have to go for the flagella, one. Here asking question is a not locomotory structure. Not locomotory structure. Directly you can go for, sir, in the same context, these are two are present now, sir, like. Now I am going to clarify this one. Both pillae and fimbriae, option 4 I am going to take. Both pillae and fimbriae are not locomotory function. Okay, should be clear. Pillae is like, for example, this one you take one bacteria. Okay, from the bacteria surface, plasma membrane only. There is a tubular structure is forming like this. This tubular structure we are calling as a pili. Okay, NCERT lo didn't mention function, but they clearly mentioned pili and fimbriae are not locomotory. Should be clear. Okay, this one is made up of special type of protein, that only mentioned. Fimbriae function is important. Okay, once a fimbriae function, attachment of bacteria to host cells. You can call the rocks or something, attachment of bacteria to the host cells. Separately function important. So function of the fimbriae is attachment of the bacteria to the rocks or attachment of bacteria to the host cell. Now I am going to clear second question, option 4. Pili and fimbriae are not locomotory structures in bacteria. Okay. Now I hope you can, with this slide, first two questions, you get the what it called as a meiosomes. What are meiosomes? What are chromatophores? Their functions. Which one are not locomotory structures? These two. That's okay. But thing is that what is the function of embryo venam? The next one I am going to make the slide. The next third question I am going to check. The following are the inclusion bodies. The following are inclusion. Yes. The following are inclusion bodies except another important in the bacteria point of view, Monera point of view, exam ki inclusion bodies. Okay. What about the inclusion bodies here? Inclusion bodies. First we speak about the inclusion bodies. Okay. These inclusion bodies are present only in the bacteria, just like a meiosomes. Okay, one. Second one is that these inclusion bodies are non-membranous structures. Non-membranous structure. Wait for the last, I will tell you. Non-membranous structures. One. Second one. These are responsible for storing of required substances. Responsible for the storing of the required substances. 3-1. Inclusion bodies are present only in the bacteria. Okay. Non-membranous structures, and they are responsible for the storing of required substances. What are the examples of inclusion bodies? Total inclusion bodies you should remember. Okay. That inclusion bodies include a phosphate granules. Phosphate granules. 1. Next one is a glycogen granules. Glycogen granules. Third one is a phosphate granules followed by the glycogen granules. Next one is cyanophycean granules. Cyanophycean granules. Next one is a gas vacuoles. These are the examples given here. Gas vacuoles in purple sulfur bacteria, green sulfur bacteria. Okay na? That one. This one. That is also important. Okay na? Gas vacuoles present in the purple sulfur bacteria, green sulfur bacteria. That is also important. Now you can check. Meiosomes 1, chromato inclusion bodies 1, some are important. Okay. Now I am going to take the following. These following are inclusion bodies except phosphate granules, gas vacuole, cyanophycean granules. I am going to answer is a chromatophores. Okay. The next point I am going to speak about this question. You may neglect, that is why I placed this one. First, fourth question, the fluid nature of the membrane is important in the function. First you have to remember, in NEET exam there is no all of the above, there is no none of the above options. Okay. For your reference purpose, without neglecting the options only, I placed the all of the above only. In the exam case, they will give A and B. Okay, don't commit immediately the answer. Okay, now you can take, first one is a fluid nature of the membrane, plasma membrane. As we know, fluid nature of the plasma membrane, that is a structure of plasma membrane is a fluid mosaic model. Fluid mosaic model proposed by Singer and Nicholson. Fluid mosaic model proposed by Singer and Nicholson. That given year is a 1972. Okay, that one is here. What it called year wise numbering is that memory is somewhat difficult only. First, just I am giving one hint is like in 1972, Singer and Nicholson, these proposed the fluid mosaic model to explain the structural and functional aspects of the plasma membrane. You understand the statement? One more, anyone can you tell anything is related to the year 1972? It's very difficult to remember, I can understand, but as a repeater point of view, we can correlate. In 1972 itself, there is a first construction of rDNA, recombinant DNA first constructed. If you remember, the first construction of rDNA by Stanley Cohen and Herbert Boyer. Stanley Cohen and Herbert Boyer are these two scientists construct the first artificial recombinant DNA. That is also happened in 1972 only. Okay, comes to the one. Fluid mosaic model, okay, that's one. Here question is the fluid nature of the membrane is important in the functions like formation of the intercellular junctions. You can understand gap junctions, tight junctions, adhering junctions, cell growth and cell division. Next one is a endocytosis. One more is there, secretion. Secretion is related to the exocytosis. Already I mentioned options will not be all of the above. Remaining everything you may read. But this one is some persons will neglect. That is why I am going for option 4. All these questions we are telling just to recollect. Very soon you are going to face. Already so many times you read the book. Okay, we recollect the information. Okay, now this one is. Most of this one you can observe. Most of the chemical studies regarding the cell membrane. Nice question, beautiful concept is there behind. Okay, question number 5. Most of the chemical studies regarding the cell membrane done by using. The question you may not analyze. Different scientists did the different experiments to know what is the structure of plasma membrane, what is the function of the plasma membrane, how the function. So to know the structural and functional aspects of plasma membrane, different scientists across the world did the different experiments. But the common thing, you know, all these scientists taken plasma membrane from one source only. Bhayankara secret. All the scientists to study about the plasma membrane, they took plasma membrane from only one source. So that question indicates most of the plasma membrane experiments, most of the plasma membrane knowledge is present this only. What is your answer? Human RBC, human WBC, frog RBC, frog WBC. Anyone? Question number 5. So here that answer is you may neglect, that is why I placed this one. Human RBC. Human RBC cells are most suitable to study about the structural and functional aspects of the plasma membrane. Frequency okay na? Human RBC are the most suitable to study structural and functional aspects of the plasma membrane. Why about the human RBC? Why not any other cell? Simple, very glorified explanation. For example, you take any cell other than the RBC. You take any cell other than the human mature RBC, other than that. This is a cell membrane, yes or no? Actually, we want to extract this only, cell membrane only we want. Okay. But the cell containing, this is another one is a nuclear membrane other than the human RBC, okay? This is a nuclear membrane. You can see endoplasmic reticulum membrane also you can see. Golgi complex membrane also you can observe. Lysosomal membrane also you can observe. Mitochondria membrane also you can observe. Understand? But among these one, which one only you require? We want to extract only the plasma membrane. But when you are extracting that one, you will get plasma membrane. But not only plasma membrane, you will get plasma membrane, cell membrane, you will get the membrane of endoplasmic reticulum, you will get the membrane of mitochondria, you will get membrane of lysosome, you will get the membrane of it. You will not get the purest form of the plasma membrane. Then from where you can get the purest form of plasma membrane to study about it? That is a RBC, human RBC. This is a plasma membrane, human RBC. Is it only plasma membrane or cell membrane? It don't have the nucleus. So nuclear membrane illa. There is no endoplasmic reticulum. There is no Golgi complex. There is no mitochondria. There is no lysosome. Nothing else. Total cytoplasm of the RBC, only membrane is a plasma membrane. Total cytoplasm is occupied by the just hemoglobin. So that only membrane in case of human RBC is occupied by only membrane is a cell membrane or plasma membrane. So the purest form of cell membrane, the purest form of plasma membrane we can extract from human RBC. That is why most of the experiments regarding the cell membrane done by using human RBC. So I hope it is, you will neglect, that is why I said explanation also. Next I am going, this is a usual question, okay na? Direct question but usual question, okay? What is the percentage of the proteins and lipids in the membrane of erythrocytes respectively? Direct question, I am not explaining. In the membranes of the RBC, that is a respectively. 52% of the proteins, 40% of the lipids. Okay? This one. Remaining as usually, this is a very important, you may get doubt. Sir, 52% of proteins, 40% of the lipids. Now, which lipids? Phospholipids. What is the example of phospholipid? Lecithin. Okay? You will get a doubt, sir, for what about the remaining 8%, sir? Remaining 8% are belongs to carbohydrates. But plasma membrane is mostly made up of the lipids and proteins. This question important, important areas only touching. Okay? Next, going to the next one. The following cell organ, this is the usual question. Okay? This is a usual. You may touch it very immediately, but usual question. Seventh question. The following cell organelles not included under the endomembranous system. Okay? So, endomembrane, I am not prolonging. Endomembranous system including endoplasmic reticulum, followed by the Golgi complex, followed by the lysosomes, lysosome, followed by the vacuole. These are 4 organelles only. G-E-R-L-V. Golgi complex, endoplasmic reticulum, lysosomes and vacuole. This system is called as a endomembrane system. Okay? Directly I am going for the option is a peroxisomes. Usually question will come. Going. Next one, 8th one. In animal cells, steroid hormone synthesis at. Anyone? In animal cells, question number 8. Which one is okay? So what it called in animal cells? Question number also can you mention? So here in animal cells, steroid hormone, konchu applicative questions I am going to discuss about it. Okay? Steroid hormone synthesis. First one, rough endoplasmic reticulum function is a site of synthesis of proteins. Site of synthesis of proteins. Idi everybody know, should know. Okay, one more point is, what is the name of rough endoplasmic reticulum in case of neuron? In case of neuron, rough endoplasmic reticulum is called as the nasal granules. Nasal granules are nothing but rough endoplasmic reticulum in case of the neurons. Okay, comes to this one. This is a site of synthesis of lipids. Site of synthesis of the steroid hormones. Directly I am going for option is a SCR. Okay. So here what is application is that first I am going to clarify steroid hormone synthesis at the smooth endoplasmic reticulum. Okay, no one. Second one, what is the meaning of steroid hormones? The hormones which are synthesized from cholesterol. 4 chapters I will connect, just you can check. First cell chapter is a steroid hormones are synthesizing at the smooth endoplasmic reticulum. One. Second one, this one is what is the meaning of steroid hormones? The hormones which are synthesizing from the cholesterol, biomolecules. What are the examples of steroid hormones? Steroid hormones include testosterone, estrogen, progesterone, aldosterone and cortisol. 5 examples. Testosterone, estrogen, progesterone, aldosterone and cortisol. 5 only examples come from the steroid hormones. Last one. It is important. For steroid hormones, receptors are present in the intracellular receptors or membrane bound receptors. Steroid hormones receptors are intracellular receptors. Intracellular receptors. Last one, 5th one. For these steroid hormones, does it involve any secondary intracellular secondary chemical messenger? There is no secondary messenger in their action. You can understand, this is a way of reading. Repeaters, this is a way of reading. Now I am going to make, okay. So these are the linked questions. I hope you are coming to close. I hope you are understanding it. Okay, different chapters na you can be. Okay, you already studied, gone through so many exams also you wrote. Okay, all chapters you have a now in your brain. So steroid hormone synthesis at the smooth endoplasmic reticulum. Steroid hormones are synthesizing from the cholesterol. Testosterone, estrogen, progesterone, aldosterone, cortisol examples of steroid hormones. For steroid hormones, receptors present in the intracellular, receptor mostly intranuclear. In the action of secondary steroid hormones, there is no involvement of intracellular secondary chemical messenger like cyclic AMP will not involve. Okay. Now, this is always important question. Okay, that is why I place a 9th question is important question directly. Okay, no explanation. That is the site of formation of the glycoproteins and glycolipids. Always so many times repeated question. So many exams, so many times repeated question. Directly I am going for the Golgi complex. Glycoproteins and glycolipids is one way of asking directly, no explanation. Second one is Golgi complex, same one. That is we are calling as a post-translational. 1 year they ask like this, post-translational modification. That is a glycosylation of proteins and lipids. Glycosylation, addition of sugar or addition of carbohydrate. Addition of sugar or addition of carbohydrate is called as a glycosylation. Important, this one. Directly, sometime majority of the cases they will ask like this only. Some cases low, they may ask, they can be, there is a possibility. There is the same option, Golgi complex involved in the post-translational modification, that is a glycosylation of the proteins and the lipids. Or average students will go wrong in this one. Okay, that is the one. Remaining functions, okay. Second one is a packing. Next one is a secretion. intracellular secretion and extracellular secretion. These are the some functions related to the Golgi complex. If question comes in the Golgi complex, this area only question will come. Remaining everything conceptual. Next one, 10th question. Which of the following cell organelle containing one time you can check 10th question. Which of the following cell organelle containing enzymes for synthesis of both carbohydrates and proteins? 10th question. Anyone? 10th question. Can you, while we are typing the options, can you check the question number also? You can place and you can type the answer, okay? Which of the following cell organelle for synthesis of both carbohydrates and protein? 10th question. Anyone? So here, 10th question. Chloroplast, Sneha, Selvan, chloroplast, anyone? C, same. Anyone? Reply. Is it mitochondria or chloroplast, endoplasmic reticulum, Golgi complex? Balmorgan, mitochondria. Mitochondria. Anyone? Tenth one is a 1. Mitochondria. You are telling about going for the mitochondria. Definitely wrong answer. Answer is a chloroplast. Direct question. Direct statement from the NCERT. Okay? Thing is that we are not studying in the conceptual way. Only wrong is that only. If you take the mitochondria, for example, directly I am telling, this is a mitochondria, just you can take this is called as a cristae, okay? Now this is a matrix, correct na? This is a matrix. What is the functions of matrix means mitochondria, if you take, this is a Krebs cycle. Inner mitochondrial membrane, electron transport system. Here both are the site of respiration. Okay, that's okay, good. Here the matrix containing there is a, here I am speaking, matrix containing a circular double-stranded DNA, okay, plus ribosomes. Which ribosomes are present? 70S ribosomes are present, okay. Along with that one, RNA is also present. So, matrix of mitochondria containing a circular double-stranded DNA, 70S ribosomes and RNA. So, when these DNA is present, ribosomes present means this is undergo transcription. Okay, so we will get mRNA. mRNA associated with the ribosomes to forms the proteins. So, matrix of mitochondria containing a protein synthesizing machinery. Overall function is this one. Comes to take the chloroplast in the similar way. In the similar way, if you take chloroplast, instead of the matrix, we are calling as a, what it called as a stroma. Same thing. This one is, I am speaking, this one. Double membranous structures, okay, I am not speaking about this one, okay. Now, this is also containing a circular double-stranded DNA, correct na? Plus ribosomes. Which ribosomes? 70S ribosomes, correct na? So, with this one is a matrix, stroma of the chloroplast also containing a protein synthesizing machinery, correct, right? Protein synthesizing machinery. So, up to here is a okay. Now, carbohydrate synthesis, sir. Chloroplast function is a photosynthesis. Photosynthesis, one is a light reactions. Another one is a dark reactions, or you can call it as a more correct is a CO2 fixation, correct na? Light reactions occurring in the thylakoid membrane. That energy which is released, that ATP and NADPH released into the stroma. Here, you can call it as a CO2 fixation, or you can call it as a dark reaction, or most commonly you can call it as a C3 pathway, correct na? Ultimately, this dark reaction results in synthesis of glucose, carbohydrate. Glucose, carbohydrate. See, we know, but we didn't understand it enough. Now I am going to forward, okay? I hope okay. This one, which of the following cell organelle containing enzymes for synthesis of both carbohydrate and protein? I am going for option 2 is a chloroplast. Simple thing, okay? So I hope okay. Next question I am going to make, okay? That is I know, okay? So this one is a NCERT. Next one, Amma. Here you can check, concentrate on the questions, Amma. Next one, the following are the functions. Here also, I said this will not give. Okay, for your total to cover the options I am giving. Okay, the following are the functions of the cytoskeleton. Cytoskeleton definition more or less important, but functions are important. Cytoskeleton involves the motility of the cytoplasm, mechanical support, maintenance of the cell shape. I am going for the option 4, but in the final exam law, these 3 options will be the different. Among these 3 options, only one option only will come. So, this cytoskeleton function is a motility, mechanical support, maintenance of the shape of the cell. Next one is, this is a best question. What is a site for active cytosynthesis of rRNA? rRNA means what? Ribosomal RNA. Ribosomal RNA you can call it as a rRNA. Anyone? 12th question. What is active? Where the site where constantly there is a synthesis of rRNA. That option, that is a 12th option, 12th question option. Anyone can you mention? 12th question. That one is a Joe. Next one, nucleolus, good. Adi, very important, okay na? That is why specifically I mentioned. Now you can understand, good. Some are telling that's good. Comes to the what is a, so direct answer is a nucleolus, very important, okay? What you is telling is a, you are telling answer, the answer, that what it called as answer is a beyond this one, okay, an explanation. Directly I am going for option is a nucleolus. Nucleolus, now you understand its importance, okay? Nucleolus where rRNA synthesis takes place. Answer is very good. Answer is very good. rRNA synthesis takes place, okay? I will correlate. This rRNA synthesis takes place means what are RNAs? Here I am speaking about nucleolus. Okay, na? I will tell you about nucleolus. Nucleolus where 28 srRNA synthesis takes place. 5.8 srRNA takes place. What it called as a 18 srRNA takes place. You understand, correlated with molecular biology? 20. E rRNA included 28 srRNA, 5.8 srRNA, 18 srRNA. Which RNA polymerase synthesizing these 28, 5.8 and 18 srRNA? Eukaryote only I am speaking. Nucleolus means at the eukaryote point of view. RNA polymerase 1 or RNA polymerase 2 or RNA polymerase 3. Synthesize this rRNAs 28, 5.8 and 18 srRNA. Anyone? That is answer is RNA polymerase 2. RNA polymerase 1, sorry. RNA polymerase 1. So, RNA polymerase 1 involves the sense of this one. This rRNA, total rRNAs we are calling as a simple word is a rRNA. This rRNA synthesis at a nucleolus 1. Next one is this rRNA plus proteins. Anybody remember? This rRNA plus proteins together called as a ribosomes. That means rRNA, nucleolus where rRNA synthesis takes place. rRNA plus proteins. How many types of protein? How many number of proteins makes the ribosomes? Anyone? rRNAs, I said, these are the rRNAs. rRNAs plus proteins to form the ribosomes. Tell me how many proteins to form the ribosome? How many proteins? Anyone? How many proteins? How many proteins? Ketrabalan, 80. Any other? Good. So, see, some persons are still thinking. It's about the 80 different types of proteins. 1 year given question, molecular biology law. Okay, so now I am going to proceed. This one, ribo. What is the function of ribosome? Ribosomes are the site of protein synthesis. Understand now this meaning? Nucleolus where rRNA synthesis takes place. rRNA is there means ribosomes are there. Ribosomes are there means protein synthesis is occurring. So that there is a relation between the nucleolus and protein synthesis. Going Amma, the important question, okay na? Important question, okay? One time, if you have any small inquiry, one time you can check. That's only, just to initiate, okay? So, we are very close to the exam, that is why I am making. Next question, sir, 13th question, sir, very frequent question. Okay, very frequent question. Find the mismatch pair. Direct answer, okay? Metacentric, centromere at the center, and it gives 2 equal arms. Okay? Next one here, just give a fraction. Give a fraction. Here, metacentric, centromere at the center, that this is a centromere, this one. 2 equal arms. This is a one arm, this is another arm, this is considered as a centromere, or you can call it as a primary construction. Okay? Next one is submetacentric, slightly away from the, it is frequently asking, okay? Submetacentric. One is one long arm, one is a short arm. Next one is a little bit confusion between acrocentric and stelocentric. Telocentric present at the end, like this it will be there. Acrocentric almost at the end. Centromere is almost at the end. One arm is extremely small, one arm is extremely long. Okay? That is it is frequently asking question. 1 year they went out of the boundary in this aspect. Most frequently they will ask this only. Okay? 1 year. Submetacentric, centromere slightly away from center. One it gives one small arm, one consider this is a small arm, this is considered as a large arm. The small arm is represented by the P arm. Small arm, this is a 1 year it went out of boundary. Long arm you can call it as a represented as a Q. Short arm represented by the P. Long arm represented by the Q. Very most frequent asking question, this one already we mentioned the function of glycocalyx. So, function of the Golgi complex. Okay. These are the questions. I hope okay. This one till now some areas only. Very important areas in the cell I mentioned. Okay. Now you can see here. Just say good. Next one. So, I hope okay. I covered some important areas, okay? Not to neglect. We are towards the end of the time durations, okay? We are close to the exam. That is why. Just to recollect the information, okay? The next one is cell cycle and cell division, okay? So, what we need to do is, okay, that's okay, okay? Next one is cell cycle and cell division. Which are the following phases of cell cycle metabolically inactive? Anyone? Which are the following phases of cell cycle metabolically inactive? First question. Direct questions only. G1 phase, G2 phase, Z0 phase, none of the above. Option 4. That's good. Oh, you are coming along with me. Okay. That's good. We are telling about the question number 1. Question number 14. Okay. One person is so many are getting correct only, but as a Ragul, R-A-G-U-L, Z0 state, Next one is Ragul G0 state, ZR, like a Z0 Sana. This I am telling. Answer is this one. Okay, for to avoid the confusion only, this I gave. Ana metabolically inactive. Indirectly means from the biochemistry point, what is a metabolically inactive means there is no synthesis of any type of protein. That is called as a metabolically inactive. If normally biologically, metabolically inactive means the cell is a dead. Cell is a dead. That is why for avoid 1% of the students or 2% of the student, when we are studying also, G1 phase metabolically active, G2 phase is a metabolically active, S phase is a metabolically active. So when we are telling G1S, G2 metabolically active, immediately we will consider G0 is a metabolically inactive. No one. Metabolic G0 is also metabolically active, it is a living property only. But only difference is Z0 state, cell is not ready to undergo cell division because it attains the differentiation and function. That is why I am going for option 4. Please avoid the confusion. G1 phase, S phase, G2 phase, G0 phases, all are metabolically active only. Okay? Next one, second question, direct question. Okay, I didn't take any twisted here. Morphology of the chromosomes are best studied. Going for the option 2 is a metaphase. By the time of metaphase, by the end of the prophase or by the time of the metaphase, chromosomal condensation is completed. Chromosomal condensation is completed by the end of prophase or the towards by the time of metaphase. That is why morphology of the chromosomes we can best study at the metaphase. Okay, direct question are important. That is why I taken and I am going forward. Good. Direct questions just to recollect the only. Okay, na? So not to tell the new things or something. Third question. The following are the significances of mitosis. The following are the significances of mitosis except to restore the nucleocytoplasmic ratio, correct? This is a beautiful thing. Next one is a cell repair, growth of multicellular organism, that significance. Option I am going for answer is a gametogenesis. Answer is a gametogenesis. Here, significant of mitosis, we need to go for the except gametogenesis. Okay? Next one is so we are going for the option, this one is a good. Okay, good. Be active, okay? Whatever we are studying is for the towards the end only, okay? That is why be active. Next one, Mendel laws of segregation is like a cumulative question. Mendel laws of segregation is observed during laws of segregation. If you know, law of segregation given the Mendel sir is a universal law of Mendel law. Mendel law. Almost there is no exception to the segregation. Anyone? Metaphase 1. What is the segregation is telling you? Remember na? The factors. If you take what it called as a 2 factors, these 2 factors separated, they enter into the different gametes, capital T, small t. During the gametogenesis, the 2 factors get separate, enter into the 2 gametes. Anyone? Fourth question. Fourth question, metaphase 1. First step of gametogenesis. Brother, who is the one? Balmurugan sir, you are going first step of mitosis is gametogenesis, mitosis. Gametogenesis occurs through the meiosis. That is overall, you are telling about the process. Okay, please. Towards the end, don't get confused at any cost. Okay? Okay. Here, Amma. Anyone, ma? This one, Mendel laws of segregation. I am speaking about the question number 4. Good, ma. So, Mendel laws of segregation is associated with a anaphase 1. Anaphase 1. The important, if they give cumulative question, Mendel law, very famous question. Mendel law of segregation is observed during the anaphase 1. During the anaphase 1, what happened? this during the metaphase 1 lo, just you can take this is a homologous chromosome, this is another homologous chromosome. These homologous chromosomes is one sister chromatid, this is a centromere, this is another sister chromatid. Okay, this is the one sister chromatid, connected sister chromatid. Okay, like this. This is a one pair of homologous chromosome, another pair of homologous chromosome, another pair of homologous chromosome. Here what happened? This is a staying on the metaphase, but when it comes to anaphase 1, here separation of the homologous chromosomes. Okay, this homologous chromosome entry into the one gamete, this is a chromosome entry into the another gamete. So separation of the homologous chromosomes. That is a anaphase 1. Important question. Like this question if they ask, how much we studied? No. How much we understand? It will reveal. Okay, going forward. Okay, next one ma. In the meiosis, this just now I said, in the meiosis, splitting of centromere. Anyone? Fifth question. In meiosis, splitting of centromere. Fifth question. Fifth question. Anyone? I will explain, brother. Ball-Morgan. Nucleocytoplasmic ratio, I will explain. Okay? Wait. Anyone? Splitting. Good ma. Some persons are mentioning is a anaphase 1. Here just anaphase 2. Sorry. Here you can check. This is these two are what it called as this is homologous chromosome homolog. These homologous chromosome contain two sister chromatid. These two sister chromatids held at the centromere. They are attached or held at the centromere. So in the anaphase 1, just a separation of homologous chromosomes only. So even this situation also, these two sister chromatid remains the same. But in case of anaphase 2, what happened? These sister chromatids will be separate. Sister chromatids will be separate. Then the sister chromatids are separating, there is a splitting of the centromere. Splitting of centromere. So separation of the homologous chromosomes occurred during the anaphase 1. Splitting of the centromere followed by separation of sister chromatids happened during the anaphase 2. Anaphase 2. Okay? Persons they remembered by heart, they may forget. If the persons understand, they will not forget. Going for next one. This is also somewhat cumulative I took. Okay? Sixth question. During the oogenesis, I hope you remember the human reproduction. During oogenesis, primary oocyte arrested in. As we know, primary oocyte arrested in the which phase? Normally when you are telling primary oocyte arrested in the prophase 1 of meiosis. Correct na? This is what we studied. Primary oocyte arrested in the prophase 1 of meiosis. In this prophase 1 consists of the leptotin, zygotin, pachytin, diplotin, diakinesis. In which phase it is arrested? Anyone? Good. Maheshwari, option 1. Next one is Shweta Sharvas. Good. Ilaikika, next one, this, good. So I am going for option 1 is a diplotin. Good. Persons correlated one is a good. Okay na? So it is normally when a person remembered during the oogenesis, primary oocyte arrested in the prophase 1 of the meiosis. Okay? While the person is studying repeatedly, prophase 1 consists of leptotin, zygotin, pachytin, diplotin, diakinesis. Among these, all of these comes under the prophase now. Where? I am going for the diplotin. Important, a cumulative question, if possible. Okay? Next one. The following is not significance. The last question we wrote, the following is a significance related to the mitosis. Here the following is not significance of the meiosis. Direct one. Conservation of the specific chromosome number in the each species. Absolute, very good function of the meiosis. Genetic variability. As we know, genetic variability very important. Okay? That is actually sexual reproduction event. Next one is a meristematic activity in plants. Path for evolution. E genetic variability giving the path for evolution. Okay, path for evolution. So I am going for option 3. Okay, just recollect. This 8th question, absolutely stunning question. Absolutely stunning question. Okay, always the match the following will come. Okay, find the mismatch. One time 8th question can come, everybody come over. 8th question come over. Find the answer. 8th question. 8th question. Give the number and tell. Option 3. 8th question. Can you mention the question number also, because I cannot make it, okay? Which one? Good. Some persons. Who is that? Kirta Balan. Okay, option 2. Anyone? Good. Option 3. Maheshwari, option 3. Anyone? Balmurugan, option 1. Anyone? Can you please make it fast? Anyone? Sir, one time you can check, sir, because I made it just to indicate, okay? Leptotin is a compaction of the chromosome, condensation or compaction of the chromosome. Is it correct, sir? Zygotin, synapsis. Can you make the, in the comment, I will give the 2 words. Can you make it to the comment? Can you do, sir? The one is called as a synapsis. Another one is a synapse. Can you make the difference between these two? Synapse and synapsis. Can you make the, under this video, make the comment, difference between the synapsis and synapse? Okay. Synapsis, clear appearance of the tetrad. Okay. Synapsis is a pairing of homologous chromosomes. Okay. I am only telling the answer. Okay. That results the formation of the bivalent or a tetrad. Pachytene, appearance of the recombination nodules. That one is a crossing over or recombination. Okay, I hope everybody remembered what I am speaking. Diplotin, dissolution of synaptonemal complex. Diakinesis, terminalization of chiasmin. Look like everything is correct. Look like everything. Only small difference. This one. Up to here is correct. A clear appearance of tetrad. Tetrad formation occurring in the zygotin. But clear appearance of tetrad is comes under the pachytene. Pachytene. I hope you are coming along with me. Okay. This synapsis results the formation of the bivalent chromosomes or tetrad. That's correct. But clear appearance of the tetrad you can observe only in the pachytene. Okay. So one time you can check. Match the following. Question 8th question is very, very important. Can I go forward? 8th question. Okay. Synapse. Good. Make it a comment. Okay. So I cannot check all the things. Okay. That's good. That's good. Next one, Amma. Next one. This one. 9th question and 10th question. 9th and 10th question. Come fast to the question. This 11th question also check. 9th, 10th, 11th. Give the number and tell. Good. Balburgan is a good. Synapse is a pairing of homologous chromosomes. Synapse is junction. Good. 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 Move on. Mention the number. 9th, 10th, 11th. This I gave intentionally the question. Give the option. 9th one is a 2. Okay. 10th. 10th. 10th one is a 1. Okay. Move on. Give the number question. 9th, 10th, 11th. I hope you are seeing the question. Sorry if I am there. Kamali. Okay. 10th question is a S phase. Okay, good. To avoid this confusion only, few are telling to avoid this question only this one, Anna, this one. This, what we are telling about the confirmation, whatever I am speaking is the end. Okay, again May 4th exam, then there is no much discussion. Okay, G1 phase, S phase, G2 phase, M phases. Okay, E phases are absolutely related to the eukaryotes only. If you take the cell cycle, that first statement itself, eukaryotes. G1, S, G2, M phases are absolutely related to the eukaryotes only. Prokaryote lo, there is no such a thing. Prokaryote undergo only by binary fission. Binary fission. Some persons will go blindly in that one. When they see DNA replication, they will go for the S phase. But one time, one fraction of movement, 1.5 minute time is there. Just spend 1 minute. Okay? DNA replication occurring, they are asked about. Usually they ask eukaryote. If they want, prokaryotes. So, if they ask eukaryotes, go for the S phase. If they ask prokaryotes, go for the before binary fission or before the fission. Okay? One time you can check. Okay? Going forward. So, I hope it will be useful. Okay? Next one is the 11th question. Anybody mentioned 11th? 11th one is a 2. Polyploidy. Anyone? What is this one? 11th question. Huh? You are telling about 11th question. 10th question 4. Oh, that's good. That's good, Maheshwari, good. 11th question 1. And I here to avoid the small confusion, this question I placed. Here maybe other question I heard or not, I am not sure. Next one, biomolecules. This one, observe me, observe me. There is a karyokinesis not followed by cytokinesis. Please understand me. Whatever we are speaking towards the end words. Karyokinesis not followed by cytokinesis. Karyokinesis means nuclear division. That leads to multinucleate, or you can call it as a xenocytic. Small difference is there. I am not speaking about it. But under the molecular biology, one statement is there after DNA replication. Failure of cell division. How many members observed? You can tell me. Failure of cell division after DNA replication. After DNA replication. Failure of cell division after DNA replication. It leads to polyploidy. Polyploidy. Most commonly polyploidy observed in the plants. Your understatement difference, if you understand, there is a particular level you are very clear about the concept. Or particular point you are clear about the concept. Karyokinesis not followed by cytokinesis. Xenocytic. Small difference is there, I am not mentioning. Failure of cell division after DNA replication leads to polyploidy. How many members? How many members know this difference? These two. Is Kerala NEET Academy is okay for the NEET coaching? It is the best for NEET coaching in Tamilnadu. Okay? Next one. What you are seeing is the best. What you are hearing is the best. Anyone? Is this E-Rund one? If you are very clear, you are successful. Means at a particular point of view, you are very clear about the questions. Man, class lo na mali jasta. Class lo na mali jasta. Now, sorry. Now, I am going to 11th question, explanation. Very important, okay? Going forward, okay? Now, next one, Amma. E-Rund question. 9, 10th, 11th, E-Rund explanation. Okay? NTA people are more intellectual, they know more subject. Okay, if they can ask, okay, it will go. Otherwise normal. NEET UG exam is very simple. Okay, now going for the next one is biomolecules I am going to make. Okay, now anyone biomolecules? First question, acid soluble fraction contains, acid soluble fraction contains, first one, this one, acid soluble fraction contain. Anyone question number mention and tell? Yeah. Anyone ma? This one, normally while we are studying, why I gave this one is or particular clarity. Okay. Normally when we are doing the acid test, okay, there is two things we will get. One is acid soluble fraction, next one is acid insoluble fraction. Normal, regularly students when they are studying normally. Acid soluble fraction containing a biomicromolecules. Their molecular weight is less than 800 Daltons, okay. Acid insoluble fraction generally or mostly containing a biomacromolecules. This is usually students will focus on it. Okay. Along with this one, during the acid test, this acid soluble fraction also containing a inorganic components. Biomolecules in the introduction page itself, koncham. Okay, just to giving the clarity. So, acid soluble fraction containing both biomicromolecules and also inorganic components. I am going for option 4. I hope we don't want any clarity. Okay, na? Question number is a 4. Going. Next one is a second question, false statement. Okay, the normal question only I made. Okay, amino acids are substituted methane, correct. Based on the nature of R group, there are many types of amino acids, correct. There are only 20 types of amino acids present in the proteins, correct. In amino acid, amino group, in amino acid, amino group and acid group are present on the different carbon. This statement is wrong. Present on the same carbon, that is called as a alpha carbon. Such amino acids are called as a alpha amino acids. Most common amino acids are alpha amino acids only. An amino acid in which both amino group and carboxyl group attaches to the same carbon. So I am going for option 2. Next one. Find the false statement about the lipids. R group is in the fatty acid 1 to 20 carbon. Arachidonic acid 20 carbon include carboxyl carbon. This option 1 year they given. Some tissues like a neural tissue having the lipid with more complex structures. Anyone? Question number 3. Question number 3. Question number 3. Question number 3, any option? Question number 3. R group in the fatty acid, arachidonic acid having anyone? You are typing about the question number 3. 3 is a 1, good. 3 is anyone? Good ma, going forward, good. So that's okay. That fatty acid R group is a 1 to 19th carbons. 1 to R group is a 1 to 19th carbons. I hope okay. Okay, remaining you can check. All options are good. I am going for answer is a false statement is a 1. Next one. This is a, sir, I gave like this one, but this area is 100% possibility, sir. Okay. About this one is a secondary metabolites. About the secondary metabolites. Okay. There is a absolutely 100% possibility getting the question from the secondary metabolites. So here directly I am asking, The following secondary metabolites are the toxins. Directly I am telling, okay na? One time you can check. Our secondary metabolites total table is required. Alkaloids, like what it called as essential oils, drugs, toxins, polymeric substances, essential oils, lectins, okay na? Now, this directly I am going for option is what it called as a toxin, that one is abrin, and one more is called as a ricin or ricin. Abrin and ricin are examples of the toxins. With regarding this fourth question I am giving, like synapse and synapses, anybody can you differentiate this one? Lectin, lecithin. Anyone? Lectin and lecithin. Any difference? Definitely. This fourth question, just I frame the question to indicate the question on the secondary metabolites. Definitely there is a question on the secondary metabolites. Okay, till now 2013 to till now, there is no question paper came without secondary metabolites. That's why. So, abrin and ricin are one table. Next one is make the comment here. What is the difference between the lectin and lecithin? Okay, what is that and what is this? That's enough. Which of the following is, this is also last year or before last year given question, okay? Which of the following is a complex homopolysaccharide? This area, homopolysaccharide area, there is a chance of one question we are getting. Okay, and one time, only one paragraph is there, one time. This one is, I am going for option 3, chitin. Chitin. Chitin is a polymer of N-acetyl glucosamine, N nitrogen, N-acetyl glucosamine, NAG we are calling NAG, N-acetyl, nitrogen containing polysaccharide, that is a N-acetyl glucosamine. Okay, so first we have to know this one, okay, polysaccharide, what are examples of homopolysaccharide? Our area, one paragraph should be very clear. So one is a starch, next one is a cellulose, next one is a glycogen, next one is a chitin, next one is a inulin. Which one is giving the blue color with iodine, which one is not giving the blue color with iodine, why starch is giving the blue color with iodine, chitin is a polymer of N-acetyl glucosamine, left end is a non-reducing end, right end is a reducing end. These are the points. Important. So this is also probability of question. Okay, going forward, okay. Next one, lecithin is a lipid, good. What about lectin? Next one is, assume the what is amino acid. This one, anybody can you check? Sixth question, Sixth question. Sixth question. Sixth question. Anyone? Assume the molecular weight. Similar question came on the sugars, if you remember. Assume the molecular weight of an amino acid is a 110 Dalton. What is the molecular weight of a peptide made up of 10 amino acids? Question number 6. Assume the molecular weight of an amino acid is 110 Dalton. What is the molecular weight of a peptide made up of 10 amino acids? Similar question came, similar question, okay. If you remember similar question, the molecular formula, this you try this question, okay, na? In between I will speak. The formula of glucose C6H12O6. What is the molecular formula of the maltose? If you see that question, you can understand why I gave it. Anyone? Question number 6. Anyone answer? Sixth question. Sir, usually students will go for like this, okay? Sir, one amino acid molecular weight 110 Daltons, okay? 10 amino acids means you can go for, this one is a 1100, this one. So, what is the question logic is, here, what it called as a option 2, 938 Daltons, okay, good. Expectation is correct, okay, good. So, normally usually molecular weight of one amino acid is a 110 Dalton. So, it is an average molecular weight, okay. 10 amino acid means we are going for this one. One point we need to realize is 10 amino acids means there are 9 peptide bonds. There are 9 peptide bonds. Each peptide bond is an example of dehydration. I said already this explanation, why I gave, okay. Here 9 peptide bond means 9 water molecule. Each molecular weight of the water is 18. Okay, so 9 into 18. So 9 into 18 you need to subtract from this one. I hope you are okay. Okay. So normally students may reach us up to here, multiplied. But 10 amino acids means 9 peptide bonds. That's so there is a release of 9 water molecule. Water molecular weight is 18. 16 plus 2, 18. So that 9 into 18 we need to subtract. So we are going for option 2. Similar question, that one and this one. That is why I inspired this question from that question. Okay. Next one is here. Statement, 7th question. Statement 1. All enzymes are proteins. 1. First statement is all enzymes are proteins. The next one statement, enzymes isolated from organism which live under high temperature are thermostable. Sir, first statement itself is wrong. Enzymes, when you turn the page, when you start with the same one option that starts with almost enzymes are proteins. 99% of the enzymes are nothing but proteins. Almost enzymes are proteins. We know the exception is a ribozyme. Some RNA molecule acts as enzyme called as a ribozyme. That one is example is a 23 srRNA. In bacteria, this enzyme responsible for the formation of the peptide bond. The next one, this you know. Enzymes isolated from the organism live under high temperature are thermostable. Yes, this statement. So, statement 1 is incorrect, statement 2 is correct. Example, we know from the PCR, tag polymerase, Dermis aquaticus polymerase, we are using the PCR. Okay, so I am going to conclude statement 1 is incorrect, statement 2 is correct. This graph, if you remember, Arrhenius star graph, okay. Here just I mentioned this indicates A indicates what? A indicates this point. I hope you can see properly. This indicates the transition state. 1 year they given question, by giving this one can you find this one? This arrow indicates activation energy without enzyme. This arrow indicates activation energy with enzyme. This indicates the transition state. This is a progress of reaction, potential energy. Okay? X-axis is a progress of reaction, Y-axis is a potential energy. One time check the graph. That is one important area in the enzymes. Okay? Just I am telling what are the areas we need to concentrate for the exam. Okay? Next one. 9th question. Find the correct equation for the nature of the enzyme's action. Look, this one here, why I gave this? This is itself important. This is I gave steps involved in enzyme action. A sequence of steps, one time you can check. Okay, sequence of steps. Here I am going for option, what is called as a 3. Okay, all are look like very close together, you need to discriminate. Okay. Next one, finds the false statement about factors affecting enzyme activity. False. Wrong statement about the factors affecting. Check. Enzyme function in the narrow range of temperature and pH, correct. Enzymatic activity declines below and above the optimal value. If you remember, this is a graph. If you remember, this is a graph. This is optimal temperature and optimal pH. This is a maximal velocity. Okay? Okay? This one. At low temperature and high temperature, enzymes destroy by denaturation. Third one. Km is a substrate concentration. Here you can see this like this. You can see like this. This is a Km value. This is a Vmax by 2. Is a substrate concentration where enzyme giving half of the Vmax. Almost very closely linked options. Okay? Enzymes function in the narrow range of temperature and pH. Enzymatic activity declines below and above the optimal value. At low temperature and high temperature, enzymes undergo denaturation. Good. Next one, either wrong. Option 3 is wrong. Because at a high temperature, enzymes undergo denaturation, correct. But at low temperatures, enzymes are inactive. Low active. Not at low temperature, enzymes will not undergo denaturation. You understand the difference of giving the option. At low temperature, enzymes are just inactive. Okay? They are low active. They will not undergo denaturation. They will undergo denaturation only at a high temperature. Okay? Next one. There is always the best question, this one. In the competitive enzyme inhibition law, this is the best option. Direct answer. Okay? Competitive inhibitor of enzyme succinate dehydrogenase. Direct answer is a malonate. Where is the best example for competitive enzyme inhibition, sir? This example. So, succinate dehydrogenase, you know, this is a very good exam. So, because you are repeaters, directly I am telling, succinate dehydrogenase enzyme, which is mediating the reaction, succinate to the fumarate. This one is here there is a formation of FADH2 is a forming, 1. Succinate dehydrogenase is also called as a complex 2 in the electron transport system. Succinate dehydrogenase is a common enzyme. It's the same enzyme. It's a common enzyme. It is involving in both Krebs cycle and also electron transport chain. In case of Krebs cycle, we are calling this is succinate dehydrogenase. In case of electron transport system, we are calling as a complex 2. Example is absolutely important, sir. Malonate. Different ways they will ask. This question in different ways they asked. They may ask. They will ask. If they come direct question, there is no problem for us. You are happy, we are happy, everyone happy. If they ask, just twist it. That's enough. The organic compound, this area, last area also important. The organic compound which are tightly bound to the E area lo, what is the definition of this? Directly I am going for prosthetic group. Directly I am going for the prosthetic group. Okay? In this area, one time you can check. Okay? What is the prosthetic group? Example. Heme is the prosthetic group of catalases and peroxidase, 1. The next one is the cofactors. NAD and NADP plus. Different definition you should. And last one is a metal ion. Zinc is required for the enzymatic activity of carboxypeptidase. This area, one area is just important. That's all only. Okay. So all are NCERT only. I hope it is helping. It will help to everyone. Okay. Sir, I am going to stop, sir. Karthik, sir. Okay. So I hope the question we are at towards the end of that one. We concentrate more on the questions with a little bit connections among the different chapters. Okay. Okay. So anybody having any clarity among these concepts, you can ask. Okay. That Balmorgan, anyone is having any other group, any other doubt or something, you can ask. So, So I hope everybody is preparing the best way. Okay, we are very close. Today is 30th April. Okay, 1, 2, 3. So 3 days, 3.5 days. So May 4th, afternoon 2 o'clock, you will be there. 1:30 onwards, you will be in the exam hall. Okay, those who are seriously prepared. Okay. Yes, the Selvan. Can you type your doubt, brother? So one person asked the doubt, I am going to clear, then we can wind up. Okay. So what is the meaning of nucleocytoplasmic ratio? Nucleocytoplasmic ratio. When you are studying significance of the mitosis law, what is that one we are going? Okay. Materials to be packed. Just I am reading the one somebody said. Materials to be packed in the endoplasmic reticulum, fuse with Golgi complex. Is it correct to say technically that? Not very clear. One second. Oof. That's material to be packed in the endoplasmic reticulum, fuses with a cis phase of Golgi body. Is it correct to say technically that protein and lipid synthesis complete? Here, sorry, here the point is materials, person expressed the doubt is materials to be packed in the endoplasmic reticulum, fuses with a Golgi complex. Yes. Is it correct to say technically that protein and lipid synthesis completed? Sir, here is a highly conceptual in this one law. As we know here, assume this is a cell. This is a cell ma. Okay. Assume is the nucleus, is the endoplasmic reticulum. Here the thing is that here consider this is a rough endoplasmic reticulum. Here protein synthesis here will takes place. After synthesizing the protein, the linear sequence as primary structure, that is released into the lumen of endoplasmic reticulum. Okay. So here primary structure, then convert into secondary structure, convert into tertiary structure. These, even though it is a tertiary structure, sir, the protein synthesized at a rough endoplasmic synthesis complete. But the protein is not functional. Protein synthesis complete at endoplasmic reticulum, rough endoplasmic reticulum. Lipids also synthesized, okay. But they are not functional. Then they will go and fuses with a Golgi complex. Here Golgi complex post-translational modification will take place. Post-translation means the modification occur after translation. Post-translational modification like glycosylation. You are read the what it called as a insulin law, removing of the C peptide. Those comes under the post-translational modifications occur in the Golgi. Once after post-translational modifications occur in the Golgi complex only, the protein will become a functional. That is why. So, protein synthesis will complete at endoplasmic reticulum. They become mature. They become functional after the work of Golgi complex. I hope okay. Okay, ma? So, that one cell one. Okay, one time you can check ma, that one. You asked a very good question actually, okay. So anyhow we will meet again, okay na. So all the best to everyone. So prepare the best way, okay na. We are hoping everything the best. Okay, everybody will meet, okay. Thank you very much. Good night.